Okay, so let's see how other non-carbohydrate compounds are respired. And first things first, we shall start with lipids or fats. Let's see, because you are aware that in periods of starvation, not even starvation, when you've respired glucose and you're done with the glucose, then the body can resort to fats. Okay? And you respire fats. And right from your chemicals of life, you are aware that fats do yield a lot of energy compared to carbohydrates. If you consider a given fat of a given mass and another carbohydrate of the same mass, they will yield the fats will yield more energy than carbohydrates. Okay? So before that, how are these fats then going to be respired? How are they going to be integrated in the main glucose metabolism? So the first thing that happens, you hydrolyze fats. Okay? Because a fat or a lipid, they are usually triglycerides. Okay? Triglycerides that clog together to form a fat. Now what you do is you first hydrolyze this triglyceride into a fatty acid, and this is a fatty acid. It is a long hydrocarbon chain of a carboxylic acid, okay, and a glycerol. 3 fatty acids, of course, and 1 glycerol molecule. Okay, so this is OH, and this is OH, and this is another OH. Then this is H, the rest is H. The rest, okay, H. So you hydrolyze the fat into the fatty acid, the fatty acids and the glycerol molecule. Okay, that's the first step. Now after hydrolyzing, then these fatty acids, they will be broken down into small segments, okay, of 2 carbon chain, okay, and each segment they will be fed into the metabolism or the main metabolism using the enzyme CoA, okay, where it will combine with that enzyme to form acetyl CoA, and then the CoA will combine. You remember, after forming acetyl CoA, it will be fed directly into the Krebs cycle by combining that acetyl group with the oxalacetic acid, and the pathway and the cycle continues. How about the glycerol? Now for the glycerol, you're going to phosphorylate it. You add a phosphate group, okay, on the glycerol. So when you add a phosphate group, and then after you dehydrogenate using NAD, the NAD is going to take away 1 hydrogen, and then you phosphorylate it. So when you phosphorate our glycerol, what is it converted to? It is, you convert it to glyceraldehyde 3 phosphate. Your glycerol may be enemy. So you have the OH here, and then the OH here, and another OH here. So the carbon 3, this is 1, 2, 3. You're going to add a phosphate group. This is phosphoric acid by condensation. Okay? And then after adding a phosphate group, then you oxidize. You take away this hydrogen, okay? And then you oxidize this fat so that you form glyceraldehyde. It is an aldehyde. In fact, it is on carbon 1 because it's an aldehyde. So this carbon 1, we shall take away the hydrogen, okay? And it remains as an aldehyde. Are we together? And so that aldehyde is glyceraldehyde 3 phosphate. Now after you forming your glyceraldehyde 3 phosphate, What does that mean? You remember after when you split in glycolysis? You remember when you split a glucose molecule into two 3 carbon sugars, 3 carbon compounds, the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate? Yes. Now this is your glyceraldehyde 3-phosphate. Okay? So now after forming it from a glycerol, it will continue in glycolysis, and energy will be harvested from a glycerol molecule. Okay? And it proceeds normally. 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 Is that okay? Yes. But for fatty acids, like I told you, the net yield is 19 molecules of ATP from oxidation of a triose sugar. And the NAD. Why 19? Because remember, for glucose, they are 38. But after splitting, there are 3 molecules. Sorry, there are 2 molecules that are running. But now this one is just a glyceraldehyde that is formed from one fat. So what does that mean? It will just be 38 by 2, which is 19 molecules of ATP that will be formed. Now for the fatty acids, like I told you, for fatty acids, the fatty acid component is progressively broken down, okay, into in the matrix of the mitochondrion into small fragments of the 2 carbon compounds, the acetyl groups. Okay, the acetyl groups. And then the acetyl groups, they will combine with coenzyme A to form acetyl CoA. And then the acetyl CoA will combine with oxaloacetic acid to form what we call citric acid. And the cycle continues. And you produce your ATP by substrate level phosphorylation, and you produce the reduced NAD, okay, the reduced NAD, you produce the reduced NAD, and you also produce the reduced FAD, okay, and now these two, they proceed to the electron transport system, and you harvest energy out of them, okay, as simple as that. Is that okay? Any questions for me? So that is the fat metabolism. And like I said, the way we have integrated these fats into our respiratory cycles, glycolysis and then Krebs cycle. If we want to synthesize even fats out of glucose, they are the same cycles that are reversed. Okay? If you want to convert glucose into fats, into glycol, you break it down, you form glyceraldehyde 3-phosphate, and then you dephosphorylate it, you remove a phosphate group, okay, to form a glycerol, sorry, to form the OH, and then after you reduce, okay, using NAD, reduced NAD, to form a glycerol. Now the same cycles, the same processes here, they will be reversed to form these long chains of fatty acids, okay, and now they are the fatty acids, they will combine with the glycerol, and you have your fats. And then when you want to break down the fats, the same processes are reversed, and you have your energy. So that's how your body is smart. Your body is as smart, smarter than you think, and you understand more in details when you do biochemistry. Okay? It's when you will enjoy these metabolic processes, these different metabolic processes. So now how about proteins? Remember when we were studying Krebs cycle, when we said alpha ketoglutarate, I told you to keep it in mind. I hope you did. I hope you kept it in your mind. So now this is the right time to retrieve it. Okay? So if you retrieve it, do you know why I told you that actually the whole of this respiration, right from glycolysis up to Krebs cycle, all those intermediates, they can act in quotes as links to protein metabolism. All those intermediates, talk about alpha ketoglutarate, talk about succinate, talk about malate, all those intermediates, they can act as links to protein metabolism. And with protein metabolism, it occurs when your body is starved. What does that mean? You have used all the glucose, all the carbohydrates, and they are done. You have used all the fats, and they are done. Maybe they are not done, but what is remaining is essential for other body functions. Because fats have other secondary functions, other functions. Then what do? Then you resort to proteins. So that's when you see somebody shrinking, somebody becoming tiny and tiny and tiny and tiny, to the extent of counting number of ribs. In English, they describe such a person as a bag of bones. Okay? Yes. So, how are these proteins going to be integrated into our usual metabolism, into our glucose metabolism? You need to have an idea. So, the first thing that happens is, a protein is a complex thing. So complex. Okay? So now the protein, which is complex, it is going to be hydrolyzed into individual amino acids. Okay? So the protein is hydrolyzed. Most of you will be insinuated to say broken down. Don't use this word. Breaking down, it's like you have this and you get your pan and you get your hammer and crack and each goes. That's not how it is. This reaction, it's a hydrolysis reaction where water participates. Okay? It cannot occur without water molecules. So the protein is hydrolyzed into individual amino acids. Then, after you've formed your individual amino acids, after forming these amino acids, an amino acid, maybe just a brief reminder, it's like this, NH2, okay? And then you have C, you have R here, okay? And then you have COOH. In that, if I try to do this, I do this, and then I put the amino acid here, the amino group here, it is the same thing. And then I put the COOH. So if I remove this amino group, are you seeing? And maybe I add a keto group on this carbon atom, and then I reduce this. Or even if I don't add, but if I remove this amino group, okay, and I add a keto group, and it becomes something like this. See, then you have O here. Then, and I reduce this maybe to this. Okay? How am I going to reduce? I have reduced NAD inside. So it is the one which is going to reduce the carboxyl group. Or even, I don't need to even reduce the carboxyl group, because they are even intermediates that have a carboxyl group. So, but if I do this, you see that, are you seeing that I'm forming a carbohydrate? Are you seeing that? But also if I want a protein, if I want a given amino acid, you have essential amino acids and non-essential amino acids. So if I want an amino acid and I remove this, okay, and I add this amino group on that carbon, You see that I have an amino acid, and then I oxidize this. You see that I have an amino acid. Yes. So these processes, these ones where you transfer an amino group from one compound to another, is called transamination. Okay? Transamination. So I'm going to remove this amino group and add a keto group. Okay? And then this ammonia, I convert it into urea. Okay? You read about the ornithine cycle. I convert it into urea. Okay? And then I excrete this urea. Now, the compound formed, okay? This compound, depending on the R, okay? Depending on how many carbon atoms does that compound have, it will determine which intermediate is going to be formed. And when you form that intermediate, then the intermediate will proceed in the Krebs cycle. Does it make sense? For instance, let's see. If the compound formed, okay, the residual carbon compound, a keto acid, okay, is then integrated in the respiratory pathway depending on the number of the carbon atoms that compound has. If the compound has 5 carbon atoms, like the amino acid glutamate, this one, this is an amino acid, okay? So when you remove the amino group and you add a keto group, then you form a 5 carbon compound, and that carbon compound will be converted to alpha ketoglutarate. And when you form alpha ketoglutarate, then you proceed with Krebs cycle and you produce the NADs and you produce the FAD and the ATP. Okay? And the NAD and FAD, they will be taken to where? To the ETS. Okay? For further oxidation. Okay? If it's a 4 carbon compound after removing the amino group, for example, this amino acid, aspartate, then the aspartate can be converted to OAA, oxaloacetic acid, and you know OAA. Then the OAA will combine with acetyl group from acetyl-CoA to form that citric acid, and the cycle will continue. And you produce NAD, you produce FAD, they are taken to ETS, boom, you produce energy. If it is aniline, so the alanine, which has 3 carbon atoms, then the aniline to be converted to pyruvic acid. Okay? You remember the pyruvic acid. It has 3 carbon atoms, where this is OOH, okay? And then this is OHO. And then finally this is. So you easily convert to pyruvic acid, pyruvate. And then the pyruvate will now be converted into acetyl. It is decarboxylated, okay? It undergoes oxidative decarboxylation, okay? Where it loses carbon dioxide and then loses hydrogen, okay? And when you lose the hydrogen is taken up by the NAD together with the electrons to form reduced NAD. And then the acetyl group will now combine with the CoA to form the acetyl-CoA, okay? Now this acetyl-CoA, it will now be taken to the Krebs cycle, where it will give this acetyl group to the oxaloacetic acid, to OAA, and the cycle continues. Okay? Yes, so that's basically how proteins, that's basically what happens when you are starved. And because the body is undergoing gluconeogenesis, okay, where you're producing where you're metabolizing proteins, many of them, then you produce a lot of ketone bodies. That at this stage, if you try to, if somebody is metabolizing proteins and you try to smell eh, somebody's breath, somebody will have that sweet fruity smell. Okay? Because somebody is producing a lot of ketone bodies. Then the amino acids with very many carbon atoms are then converted by transamination into 3, 4, 5 carbon atoms, and they are integrated within metabolism using these processes, these three. And you have energy out of proteins. Is that okay? Yes. Any question for me? Yes. So basically that's how proteins are respired, since there are no questions for me. So now let's check how other carbohydrates are respired, and this is just simple. For example, fructose. I think even the body prefers respiring fructose than glucose. Because remember, with glucose, you first phosphorylate it, and then after phosphorylating glucose, then you isomerate it, or you isomerize it to form fructose 6-phosphate, and then you phosphorylate the fructose again. But now here, you already have the fructose. So it is present in many free forms and in many fruits, and also formed by hydrolysis of sucrose in the ileum. So this is fructose. This is just nutrition. This is senior to science. Okay? So now, in muscles and kidneys, you're going to phosphorylate the fructose to form this. Okay? By this enzyme, hexokinase enzyme. And then the fructose 6-phosphate is then going to be phosphorylated again to form fructose 1,6-diphosphate, and then you split it. And after splitting it, you form the two molecules of glycerolide 3 phosphate, and then dihydroxyacetone phosphate. That one happens in the muscles and kidneys. But in the liver, okay, the fructokinase enzyme, it phosphorylates the fructose to fructose 1,phosphate. Okay? And when the fructose 1,phosphate formed, now in the liver it will split into glyceraldehyde and then the hydroxyacetone phosphate. Then the glyceraldehyde is going to be phosphorylated again. This one is going to be phosphorylated to form glyceraldehyde 3-phosphate, which will now proceed in glycolysis. Okay? In glycolysis. And then the process continues. Respiration continues. The dihydroxyacetone phosphate, it converts. We almost talked about this. Okay? We talked about this. And I hope everything is okay. If glycolysis is okay, then here is okay. So that's why I say that I think the body prefers, okay, to respire fructose to glucose, because this one is immediate, no isomerism. You phosphorylate it again, and it splits there and then to give you the 3 carbon compounds. But for glucose, you phosphorylate it and then you isomerize it to form the fructose 1,6-phosphate. Okay? And then the fructose 1, you phosphorylate it again to form, and so on, and the cycle continues. Now for other hexose sugars such as galactose, for instance, galactose is the same, is identical as glucose, but just that the difference is on carbon number. Is it 2 or 4? One of the two. The difference is on carbon number, either 2 or 4. Okay, but the rest is the same. So if you can respire glucose, then there is nothing which stops you from respiring galactose. So now, galactose is first phosphorylated to form galactose 1,6 phosphate and then converted to fructose 1 phosphate through a series of different enzymatic reactions. Now, after converting glucose 1 phosphate, then the rest becomes the same. Okay? The rest becomes the same. Where the glucose now is going to isomerize to give you the fructose 1 phosphate, and then you phosphorylate it to form fructose 1,6 diphosphate, and then the diphosphate being a high energy compound, then it will split to give you the 3 carbon compounds. So it's as simple as that. Then for mannoses, these ones they are released in digestion of various polysaccharides and glycoproteins of food. And you can also phosphorate them at this carbon 6 using hexokinase. And when you phosphorylate, then it isomerizes to form glucose 6, glucose 1, and then you phosphorylate it again to form the fructose 1,6 diphosphate. So all these ones they are disaccharides, and with disaccharides, for them they are not as complex, okay, as proteins and the fats, the fellow counterparts fats. Okay, yes, it doesn't involve a lot of metabolism. It doesn't involve a lot of steps, and it's as simple. So I want you to have a look at this. Then mannose 6 then isomerizes to form fructose 6, and then the fructose 6 is phosphorylated to form fructose 1,6, and then the cycle continues. Now I want you to have a look at this. So you can see. So this is a summary of metabolism. Where this one, it is the main respiratory cycle. Okay? This one is our main respiratory cycle. Now where you are seeing these ones, those are the key points. Okay? Where these other non-carbohydrate compounds can be integrated from. For example, if they are polysaccharides such as starch, glycogen, cellulose, hemicellulose, they'll be broken down. They'll be hydrolyzed to form a hexose sugar. And in the hexose sugar, then it will be phosphorylated to form a hexose phosphate sugar, which now enters into the cycle. If they are fatty acids, if they are fats, the fats is going to be hydrolyzed to form a glycerol and fatty acids, as you can see. Then the glycerol is going to be oxidized, okay, and then you phosphorylate it. So in other words, it undergoes oxidative phosphorylation to form a triose sugar, which is phosphoglyceraldehyde, and then it continues. Then for the fatty acids, they will be broken down into small fragments of 2 carbon compounds. Okay, the acetyl groups, and then these acetyl groups, they will combine with the acetyl CoA. Okay, they will combine with CoA to form what we call acetyl CoA. And now this acetyl CoA is the one that will take this fatty acid, this small fragment, into the Krebs cycle, and you produce energy out of it. But if it's a protein, then the protein will be hydrolyzed, like how you can see, okay, to form amino acids. Now, depending on the amino acid, it will determine which stage is going to be integrated from. If it is aniline, Adenine, which is a 3-carbon compound, then it will be converted to pyruvic acid. If it is aspartic acid, which is a 4-carbon compound, then it will be converted to oxaloacetic acid. If it's glutamic acid, which is a 5-carbon compound, then it will be converted to alpha ketoglutarate. But at each stage, where you can convert this to this, you can also convert the same back. So these are reversible reactions, like I said. So if you want to also form proteins, you undergo respiration, produce these intermediate products, and then these intermediate products, they are the ones that will be converted into the individual amino acids of your choice. If you want fats, maybe there's positive energy balance. You have too much carbohydrates than what your body needs. Then you form that glyceraldehyde, which is going to be an intermediate product in formation of almost all these things. Okay? Because imagine plants, the process of photosynthesis, you produce phosphoglyceraldehyde, PGAL. Okay? Phosphoglyceraldehyde. Now it is this phosphoglyceraldehyde, it is a 3-carbon compound. So it is the one which will condense with another PGAL, and then you dephosphorylate to form the glucose. But since you are not interested in only glucose, then the phosphoglyceraldehyde that is formed within plants by photosynthesis, it is a precursor for formation of diverse ranges of compounds. You want to produce amino acids? PGAL. You want to produce fats? You the plant, PGAL. You want to produce polysaccharides? PGAL. Okay? So these integrations, all these reactions, they are reversible, and you can form anything out of this. And that's how our bodies are able to do that. Okay? Yes. So I want you to have a look at this just to appreciate how your body is smart. The picture that is coming, this one, this one. So that is the metabolism in your body. That's the metabolism in your body. And the blue one, this one, that's the main respiratory metabolism. This one. The rest, they are and they are all integrated. Okay? They are all integrated. If you want a nucleotide, you can form it from a glucose, from a carbohydrate. If you want an amino acid, you can form it. If you want a lipid, or if you want to hydrolyze liquids to produce energy, you can still do it. If you want the metabolism of cofactors and vitamins, yes. If you want vitamins, there is a point where they will integrate. And at that point, that's where you get the intermediate, which you will use to synthesize a vitamin. So, how is this? Is it too much? No, you're not supposed to know this now. But I assure you, when you do biochemistry, you will know all this. You will be able to spit all this. Any questions for me? Yeah, since nobody has a question, then it's okay. It's okay. I hope things are going on well. Okay. So let's see how respiration is controlled. And I want you to think of factors. You are the ones who are going to discuss this. Think of factors that do affect the rate of respiration. And then how is it controlled? So now, respiration, you produce energy, like I told you. And energy is nothing you cannot play along with. It is hard to produce. So, once you have it, then you should not waste it. And you should not waste your time, okay, in generating what is not needed. So, because of that, then the rate of respiration can be controlled. So because the main function of respiration is produce energy, ATP, it must be regulated so that the ATP generated only when it is needed. Don't just produce. Don't be luxurious. Don't be extravagant. And you produce when you don't need. So you must ensure that you produce only when you need. So this occurs in a number of ways. Number one, at the cellular level, and then at the organism level. And at the cellular level, the rate at which respiration occurs is regulated by the state of energy in a cell. Depending on how much energy do you have in a cell, it will determine how much ATP, how much rate of respiration is going to proceed. Okay? The ratio of ATP to ADP in a cell. Okay? And here, these two, but especially ATP, it works as an inhibitor. And to be specific, a negative. So this is negative inhibition pathway. So can somebody prepare to tell us what this means? How is ATP production going to affect the rate of respiration? And what is negative feedback pathway? Can somebody give it a shot? Okay, now I was saying you cannot keep the rate of respiration high. Okay? Imagine when you are sleeping and when you are playing. Should the rate of respiration be equal in those two states? When you are sleeping, should the rate of respiration be high? How about when you're exercising? Should the rate of respiration be high? And the answer is no. Okay? So the rate is going to be regulated, is going to be determined by the amount of ATP and amount of ADP in a cell. Okay? And how do they do it? By negative feedback. Let me do this. Negative feedback mechanism. Okay? Where they will regulate these enzymes that are involved in respiration. And we are going to see how. I hope it's what is coming. Now, before this, this is, let me first, before this. Now, what is feedback mechanism? Let's say you have your chemical reaction that involves the different steps. A, then B, and then B converted to C, and then C converted to D, and then D finally converted to the final product E. Okay? So you have A is converted to B is converted to C, and C is converted to D, and finally D, and this is the end product. Okay? End product. Now, what does this mean? If I want to regulate the rate at which E is formed, actually all these ones, they are enzymes, and this is enzyme 1, this is enzyme 2, okay, and this is enzyme 3, and finally this is enzyme 4. Now, if I want to regulate the rate at which this E is produced, if I remove one product, maybe B, E is not going to be produced. If I remove C as the intermediate, E is not going to be produced, but B is going to be produced. If I remove D, E is not going to be produced, but C is produced and D is produced. So now if you remove this, where would you put C that is accumulating? If you remove C, where would you put B that is accumulating? But if you remove B, these ones, they want to be produced, but also your starting point, which is a reactant, you can convert it into another form, or you can store it. Okay? So the best way to control this reaction is by removing the B. Now with negative feedback mechanism, or with negative feedback, the product that is formed, okay? This is our product that is formed. It can inhibit this enzyme E1 that controls the very first step. Okay? Let me repeat. The product that is formed here in this, when a chemical reaction is determined by very many steps, more than one steps, and all these steps are controlled by enzymes, then the product that is formed in this chemical reaction, it will inhibit, or it has that ability to inhibit this enzyme E1 that can catalyze this very fast reaction. And so when you inhibit this enzyme E1, you won't form B. No B is going to be formed. And if no B, then no C. And if no C, then no D. And if no D, then no E is going to be formed. But however, that happens when E is in excess, when you have much of E. But if the concentration of E reduces, okay, then the E that had attached on this enzyme, that had inhibited on this enzyme, is going to go away. And then this enzyme will catalyze this very fast reaction and you form B, C, D, and finally E. So that is negative feedback. When a product formed has ability to inhibit the enzyme that is controlling the first step. And so when you inhibit that enzyme, then you will inhibit the whole rate of respiration. So what does that mean? Let's see how how the levels of ATP and ADP are bringing about the control of glucose. So right from that above principle, if the high levels of ATP in a cell, or in a cell if you have there high levels of ATP, then the ATP is going to inhibit this enzyme, hexokinase. And now this is the enzyme that can phosphorylate a glucose molecule directly when it enters into a cell. So if this enzyme is not stimulated, okay, if this enzyme, the hexokinase, is inhibited, then it means the glucose is not going to be phosphorylated. And if you don't phosphorylate the glucose, then what does that mean? The whole of respiration is going to be cut off. And so the cell will use the ATP that is available. Okay? Remember, when you use ATP, it's ATP. You use it by removing an organic phosphate, okay? So that you form ADP. Now it means as you are using ATP, you produce more of ADP, adenosine diphosphate. Now if the high levels of ADP, or if the levels of ADP increase in a cell, then our enzyme hexokinase is going to be stimulated. And if the enzyme is stimulated, then the glucose molecule is going to be phosphorylated. And if you phosphorylate the glucose, then what will happen? The whole of the process is going to take place. And finally, you produce ATP. Okay? Then ATP increases again. Then this enzyme inhibited. then you inhibit the whole process of respiration so that no ATP is produced. The level goes back down, and this one goes high, and this one. So the cycle keeps on increasing, and in that, you'll be able to regulate the process of respiration. Is that okay to all of us? Yes, so basically that's what we are talking about. So high active cells, the liver cells, muscle cells, and sperm cells, they do utilize ATP very fast. Okay? They do utilize ATP very fast. And so the ATP does not accumulate in the cell. Okay? And so if the ATP does not accumulate in the cell, then the cells cannot be inhibited. So the respiration will always be high. Okay? The rate of respiration will always be high. Not so in such highly active cells. The liver cells, the muscle cells, and the sperm cells. By the way, are you aware that sperm cells do respire fructose? Think about why, from what we have just discussed. So the sperm cells do respire fructose. So find out why. Okay? Yes. Guess there is a question? Maybe. So that is. So how about for the whole organism? Now for the whole organism, okay? Where is my pen? For the whole organism, the respiratory rate or respiration rate is going to be influenced by the environmental factors. And the factor 1 is temperature. Factor 2, body size. Okay? And this one has something to do with temperature. And then finally, the physiology of an organism. Maybe when somebody's under a state of physical exercise, somebody's growing, or a seed is dormant, a dormant seed, you cannot compare the respiration rate of a seed that is dormant and a seed that is germinating. Those two have different physiological changes, and so they have different respiration rate. Now, temperature, you know, increase in temperature increases the rate of respiration. Okay? Where is this pen? Yeah, it's here. So generally, very low temperature slows down the rate of respiration in both homoeotherms and poikilotherms, in both warm-blooded and cold-blooded. That's what you are used to. Okay? If respiration is low, okay, if temperature is low, then the rate of respiration also becomes low. And this is just simple. But when temperature increases, okay, for increase in temperature by 10 degrees Celsius, for instance, maybe you are at 0 and you increase to 10 degrees Celsius, then the rate of respiration doubles. Okay? If it was at 5, it will double and it goes to 10. Then another 10, it still doubles. Another 10, it still doubles. Now that one is in poikilotherms. However, in homoeothermic organisms, for you can control, you produce hormones, you employ mechanisms to control. Okay? So you reach a level where increase in respiration now, it will decrease the rate of respiration will stop increasing. Why? Because you have employed different mechanisms to stop the further increase. Otherwise, if you don't employ those mechanisms, then you can respire and die. Okay? So explanation I've given. The rate is low because temperature affects the enzyme activity. Okay? And you know, there are enzymes that control this rate of respiration. And so at low temperature, then the rate of respiration also becomes low. Okay? Now, however, for homoeuthermic organisms, still the warm-blooded organisms, still for you produce hormones, okay, that do increase the rate of respiration. So, however, in homoeutherms, warm-blooded animals, there is a need to increase the rate of respiration to generate enough heat for maintaining the body temperature constant. Okay? And so the temperature being low, you don't respond in such a way. Then that's when you produce adrenaline, that's when you produce the thyroxine hormone, that's when you produce a lot of insulin hormone, and these hormones, they will forcefully increase the rate of respiration to generate enough heat to maintain the body temperature constant. Okay? However, now for these ones they gave up in cold-blooded. For them, they just see things. Their body temperature varies with the temperature of environment, and so the rate of respiration. Okay? Yes. But however, if temperature increases, okay, if it is if the environmental temperature is down, then they find other ways of surviving. They burrow, they hibernate, they they find other ways of surviving since they cannot control. Why? Because temperature decreasing, the rate of respiration decreases. Okay? But however, if temperature increases to nearly that of the body temperature, then the rate of respiration is going to increase. And if the rate of respiration increases, then the organisms become active. Okay? That's why actually mosquitoes, this fact, and flies, you cannot find them in Europe. Why not Europe? The temperature there do fluctuate a lot. In this time you have summer, then next time you have winter. This time you have summer, next time you have winter. So these organisms that cannot control their internal temperature, then they can't survive there. They will survive in tropics in Africa. That's why in Africa malaria is killer. Malaria is too much. Okay? Because the temperature favors the growth, the respiration of these small insects that cannot control their own body temperatures. Hence, they have a high rate of respiration, and so they'll be active, reproduce, multiply in numbers, and boom, they spread malaria at a terrible speed, like wildfire. Is it okay to everyone? Yes, yes, that's okay. However, if the rate of respiration now is high, beyond the optimum, beyond the norm, the rate of respiration decreases in homoeuthermic organisms, in warm-blooded organisms. Why? Because for you maintain your body temperature constant. Then if the environmental temperature increases beyond what is required, beyond normal, then that's why you employ mechanisms. You produce hormones to respond to that. And so the body employs mechanisms to regulate heat. And we have seen that respiration is one of producing heat in our bodies. So since the environmental temperature is high, then the need has shifted from temperature. Now, the temperature outside is too much than what you need. So the rate of respiration should not continue. Okay? It should cease. It should stop. It should decrease. So that you don't produce a lot of heat inside you. And so you maintain the body temperature optimal. This slide is confusing, but that's why I've taken some good time to explain point by point. I hope it is now okay to everyone. Maybe let me check the chat box. It's not recording. Sorry. So like I said, let me repeat for those ones who want to be there, who are not here. Like I said, small organisms have a large surface area to volume ratio. Okay? And large organisms have a small surface area to volume ratio. If you want to illustrate, then you can still illustrate, but that's under homeostasis. Now, these small organisms having a large surface area to volume ratio, they lose heat at a high rate, okay, compared to these these large organisms. And so what does that imply? It implies that these small organisms must have a high metabolic rate, a high rate of respiration to generate enough heat to replace this heat that is lost, okay, than their fellow large organisms. Then level of activity, same issue. Organisms that are more active, small organisms even are more active than the large organism. So organisms that are more active have a high rate of respiration than organisms that are just there, passive. That's why if you are to compare Usain Bolt with you, the rate of respiration in Bolt is high to produce that robust amount of energy to be used in sprinting. The birds, highly active. Imagine flying. Do you know how much resistance is in air? Too much resistance. And on top of that, no support. There is gravitational force. So you must have energy to overcome all that. Okay? So where are you going to get the energy from? Respiration. So they have a high metabolic rate to produce enough energy to keep them floating in air. Then growth. Yes, organisms that are under growth or that are under their growth spurts, they have high rate of respiration than organisms that have attained their maximum size. That's why you should feed your young ones, your kids. Okay? You should feed them really. Kids at their growth spurts really should be fed. Otherwise, if you don't feed them, then man, they will be stunted. They won't grow to their normal sizes, normal heights. Actually, that's why Africans, Americans, you always hear American height, American height, American height. They are not that tall. It's not that they have tall genes for tallness than us. Yes, we also do have. But genetics interacts with environment. You may have your genes of being tall, but if you are not fed, then you can't grow up to that size. Genetics interacts with environment. So now these Americans, for them, they know that a kid at this age or in this age bracket, usually adolescents, and then from the young ones, and then adolescents, they are in their growth spurts. They require food. They must be fed. They are growing. So you must provide them with food. Okay? And all food values, balance that. Enough amino acids, all the 20 must be there. Otherwise, if there is some amino acid that is lacking, then somebody is going to be stunted. So they provide all the 20. They provide all carbohydrates, enough. For us here, what we know is giving food, students portion. Imagine students, we give them portion beans. Anyway, portion beans, they are also good. If you don't portion beans, you have all that winter amino acids, which is good. But for them, that's not enough. They provide what is required so that somebody respires, the rate of metabolism is high, so food must be there to respire and produce energy, which is going to be used in growth. Not so. Yes, I hope that one is good. And which process? Cell division, protein synthesis, and other metabolic reactions. Then finally, I think I'm going to stop here. We have dormancy, okay? And during, we have, for example, an organism when is hibernating, or an organism when is estivating. Find out the difference. Anyway, estivation means that state of body, that state of inactivity during summer, and hibernation, it is that state of inactivity during winter. So organisms in these two states, they have low metabolism, low metabolic rate. Why? Because the environmental conditions are not favorable. So you try to hibernate, you try to estivate, try to hide, okay? So that you can survive that extreme environmental condition, okay? Yes. So respiratory rate is always low to avoid depleting the food reserves before the unfavorable season ends. So basically, that's it. So next time, I think we shall begin from respiratory quotient and how we can use the RQ values to tell which type of respiration, what is being respired, and so on and so on. And I assure you, today I wanted to finish the topic, but yeah, it was too much. But I assure you next time we shall finish it, inshallah.